Hello everyone, thanks for attending. Today I'm going to give you my presentation. It's about ontology-based collection and analysis of natural and live animal hosts of human coronaviruses. My name is Wang. I come from Guizhou University of China. My major is medical information engineering. Now I studied in University of Michigan Medical School. Thank you, my mentor, Dr. Yongxun He. He helps me to prepare the abstract and the slides. Thanks a lot. Coronaviruses are a large family of viruses, which cause the zoonotic disease. Two human coronaviruses disease, including SARS and MERS, have caused major losses in the last 2 decades. SARS-CoV was responsible for an outbreak in 2002-2003. This outbreak affected at least 8,000 individuals and caused a case fatality rate of approximately 10%. SARS-CoV has been acquired by humans from carnivorous wildlife animals such as civet cats, which are thought to from rhizophid bats. The MERS curve was discovered in a fatal human case of pneumonia in Saudi Arabia in 2012. The case fatality ranged around 35%. It's isolated from dromedary camels. Except human coronavirus, other animal curves also include important livestock pathogens, such as transmissible gastroenteritis virus of swine, bovine cough, and feline coronavirus. As the COVID-19 pandemic from January 2020, there are some available trackers and websites to it. In Johns Hopkins website, we can find that as of 8 p.m. Eastern Time, June 29, there are more than 10 million people infected COVID-19 worldwide, and more than 500,000 people died. It gave us a devastation. So finding the origin of SARS-CoV-2 will be great help to the future's mania, infectious disease prevention and control. In January 2020, scientists deciphered the genome of SARS-CoV-2. It came from a 41-year-old man. The man worked at the seafood and wildlife market in Wuhan, where a variety of mammalian species were available for purchase at the time of the outbreak. The photographs were taken when the authors visited the market in 2014. From the figure, we can see some of the wildlife on sale. It may provide a patent mechanism for zoonotic transmission. As we all know, from January 2020, there are so many researches on the potential host of COVID-19. There are some of animals have been experiment found or just through computationally predicted, just like the snake, the mink, the bats, and the penguins. Also, they found that the ferrets and cats efficiently can replicate the SARS-CoV-2. Also in May 1, the scientists used SARS-CoV-2 spike protein predicted to mammals to can infected COVID-19, but not fish, birds, and reptiles. So we think about that. hosts can be infected by human coronaviruses in natural or laboratory environments, and through ontology-based connection and analysis of known hosts of human coronaviruses can we found some potential natural and intermediate animal hosts for SARS-CoV-2. We have connected articles to identify various hosts of human coronaviruses, including laboratory animal models. All the viruses and hosts were classified using the ISCB taxon ontology, Ontofox, and Protegia, our editor. Additional axioms and terms were added in CIDO to link the coronaviruses and their hosts. In the figure, we can see the taxonomical classification of the 7 human coronaviruses and the even infectious bronchitis virus, murine hepatitis virus. Basically, coronaviruses are positive strained RNA viruses belonging to the order Idovirials, the family Coronaviridae, and the subfamily Coronavirini. Contains the 4 general alpha, beta, gamma, and delta coronavirus. We all know SARS-CoV and SARS-CoV-2 are members of the genus Beta coronavirus. MERS-CoV, also under the same genus Beta coronavirus. In addition, there are 4 human coronaviruses called common cold. The 229E and NL63 belong to alpha coronavirus. HKU1 and OC43 are the lower classification of beta coronavirus. In the figure, we saw the 17 natural animal hosts of different human coronaviruses. The taxonomic relations among these hosts are provided in the figure. The 12 natural hosts of SARS-CoV, including bats, masked palm civet, Ranking dog, greater horseshoe bat, Chinese horseshoe bat, mammalian, penguin, etc. And the phylogenetic studies have already pointed the origin of SARS-CoV from in bats. Its intermediate hosts include those have been identified like masked palm civet, ranking dog. SARS-CoV also have been detected in smaller animals, for example, lesser rice-field rat, red fox, and domestic rat. And in SARS-CoV, its lateral and intermediate hosts include bats, dromedary camels, and Bactrian camels. We also know that NL63 be found to infect masked civets. Its natural host is bats, and the virus also being found in camels. Meanwhile, T2-E also isolated from alpacas raised in capacity with dromedary camels. NL63 owning the natural host bat is known, and for OC43 infected humans and cattle. HKU1 is currently suspected to be a rodent-related virus, originally obtained from infected mice. Laboratory studies of SARS and MERS over the past decade have used a number of animal models. The techniques refer to experimental animals of SARS and the underlined ones to the experimental animals of MERS. The experimental animals of SARS including the most commonly used common marmoset mouse, and African green monkey, hamsters, macaques, Syrian hamsters. Unlike SARS-CoV that can infect many small animals, MERS-CoV can't achieve infection because the receptor binding domain of the viral spiking protein DPP4 in MERS-CoV has glycosylation sites that block viral recognition. So, rhesus macaques and marmosets are currently the best MERS animal models. At present, a number of experimental animal models of COVID-19 has been used in vaccine and drug development. Many transgenic mice and macaques can infect and replicate SARS-CoV-2. Also, ferrets, cats, and golden Syrian hamsters can be experimentally infected with the virus and can spread the infection and other animals to the same species in laboratory settings. We use CIDO. It means coronavirus infectious disease ontology. It's a communicated-based ontology for coronavirus disease, including COVID-19, SARS, MERS, etc. We use related taxonomy terms extracted from ISMB taxon. Then imported to CIDO support more systemic representation and analysis. CIDO proposes a relation to link the virus and the host, which represents a relation between a microbe and an organism that serves as the host whole and can host the survival of the microbe, such as MERS-CoV has susceptible host, some commoners. Now we can talk about this discussion and conclusion. As we all know from recently news, there are more and more reports on animals being infected with COVID-19, such as minks in Dutch farm, tigers in American zoo, and some pet cats and dogs in Hong Kong. So there needs further research about mink, penguins, tiger, cat, dog can be COVID-19 potential hosts. In our next work, we want to use the annotated data to identify the other potential mammalians that the coronavirus can spread. Yes, in our research, all the natural and laboratory animal hosts we found with experimental evidence show that they all belong to mammals. But I think it needs further investigation. This is the end of my presentation. Thanks the experts Yongchun He, Yongyu Hongyu, Xian Weiye, thanks Feng Wei Zhang, and we also use the ontology tools Ontifox NCBI taxon CIDO. Thanks Guizhou University Overseas Study Plan. It supports me to study in Dr. He's lab. Thank you for your attending. Thank you for your patience. We are glad to take questions.